Nicholas, we can start. Yes. Good. So, but for the sake of the recording, I think I can quickly repeat. So just pardon me. So our guest is Professor John Demes Mande, who is our Executive Dean, Faculty of Veterinary Medicine, University of Nairobi. We have Dr. Paul Musiri, who is the head of Botany Department, National Museums of Kenya. Dr. Telessa Leonardo, who is a Director, Citizen Science and Special Projects, the National Geographical Society. And our presenters are Dr. Carlos Velansko of the National Geographical Society, and in particular, iNaturalist program, Dr. Paul Kirika from the National Museums of Kenya. He is also a senior scientist or research scientist. Then we have Dr. Mire Nyawira, again, a senior research scientist from the National Museums of Kenya. And we have Susan Kafashia, research scientist in the National Museums of Kenya. And I said myself, I'm Dr. Mushane Mushai. I work for the University of Nairobi, the Faculty of Veterinary Medicine, the Department of Clinical Studies. And section is wildlife section, where we deal with wildlife management and conservation. Otherwise, thank you very much. So I was saying that we are going to record, and the webinar is being recorded. So it will be available at least 10 minutes after the end of this webinar. It will be shared in our project YouTube, and the YouTube link is going to be in our chat section. We're also going to share it in our Facebook, or the project Facebook. Again, it will be shared by our project manager in the Facebook for the project. So all presenters are requested to put their questions in the chat section, because we are going to answer them during the question and answers section or session that will be made after the presentation are already made. So please write your questions in the chat section so that we can be able to get them from there, and we'll be able to direct them to the various presenters. Otherwise, I hope that for those of us who are continuing to joining, please feel welcome. And now I'm going to call Dr. Mire Nyawira to give us the welcoming remark. That one is going to be followed by, I wanted to share a quick one, the program for the day. Let me share quickly. Okay, 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 no. Okay, let us first and foremost, I'll share it later. Let us have Dr. Mire Nyawira starting with welcoming remarks. Welcome.
Good afternoon, everyone. For those who may not know me, I'm Dr. Mire Nyawira from National Museums of Kenya. I'm a research scientist working in fungi ecology, and also soil ecology. I take this opportunity to welcome you all to this webinar. This webinar is on engaging citizen scientists in macrofungi or in mushroom research and conservation. And I recognize the presence of our distinguished guests. I recognize the presence of our esteemed participants, and I am happy that we are here so that we can learn together. And I'm sure we are going to benefit, and I'm sure we are going to, our expectations are going to be met. In this virtual conference, I wish to recognize all the presence of our distinguished guests. In particular, I want to recognize the presence of Dr. Musiri, Poro Musiri, who is the head of Botany Department where I work, National Museums of Kenya. Welcome, Daktari. I also wish to recognize the presence of Professor John Demesi Mande. He is the Executive Dean, Faculty of Veterinary Medicine, University of Nairobi. Welcome, Professor. I'm also very grateful and humbled to welcome Dr. Telessa Leonardo. She's Director of Citizen Science Program, National Geographical Society. Welcome, Dr. Telessa. I also recognize all our participants. Please feel welcome to this webinar. We are happy to note we have participants from all over the world. And I take this opportunity to welcome you all, those from Kenya, East Africa, Africa, and all other continents. Feel welcomed to participate in this webinar, and I'm sure you are going to benefit a lot. We are also privileged in this webinar to have very experienced and very knowledgeable presenters. We have presenters from National Museums of Kenya. We have presenters from National Geographical Society, and we have presenters from University of Nairobi. So from these people who are very knowledgeable, I'm sure we are going to learn a lot. So let us wait so that we can hear from them. So we welcome you all to our presentation. The webinar is focusing on creating awareness on the importance of macrofungi. Most of us know them as mushrooms in our ecosystem, with the aim of now increasing interest, increasing knowledge, and also increasing the skills of both scientists and both citizen scientists so that they can effectively and accurately engage in mushroom research and conservation. I wish you all the best as you listen to the presentation, and I hope this webinar is going to meet all your expectations. Welcome all.
Thank you very much, Dr. Mary. So what I was telling you, I want to quickly share the program so that we are all aware what is happening. So this one. So our program today. So far, we are now in the opening remarks. So we'll have 3 opening remarks. The Professor John Mande will start because he is going for another meeting, followed by Paul Musiri and then Dr. Telessa Leonardo. And then after that, we will move on to our presenters in this order. So welcome. Now I want to stop sharing so that now I can invite Professor John Demesi Mande from the University of Nairobi, who is also the Executive Dean, Faculty of Veterinary Medicine. Professor, welcome.
Thank you very much, Dr. Good afternoon, colleagues and researchers and participants. I'm delighted really to be a part of this webinar. I was alerted about it more than a month ago when Dr. Mushai told me about it, and I was quite glad to be a part of it. Just that we have another stakeholder function that is involving leather science and technology at a postgraduate level. And I'll also just be indulging you that you allow me to go and visit virtually. But first of all, University of Nairobi is a premier university, you know, and our focus is teaching and learning. We also are keen on research. We are also very keen on community outreach. And so when Dr. Muchai and his team agreed to partner with us more than 6 years ago to teach wildlife management and conservation, we have seen a transformation in the enrollment of students. We've seen growth, and so we want to just appreciate him and his team, and also linking the university to National Geographic Society, National Museums of Kenya, and iNaturalists, and this Wildlife Conservation Society and the community of researchers. We are really grateful. Let me just say then that when growing up, during the month of February and March, you know, when the land would be cultivated, and you would really be quite excited to see when it had just rained that, you know, you'd see a mushroom growing, and so it was quite a delicacy while growing up, especially when you consumed it with ugali. I don't see it anymore because we spend most of our time in Nairobi and all that. But, you know, just to appreciate that you can actually cultivate that product and exploit it for nutrition, exploit it for medicinal purposes, and also just, you know, ensuring that you're able to tap it into all the aspects of the biodiversity. I believe that you can also use it to manage many other issues in the ecosystem. So you have individuals who have grown up, gone to school, earned PhD just to study the mushroom that I used to consume. To me, that's quite humbling. And therefore, also ensuring that you can exploit it for revenue and income. It's very interesting. Let me not go into that area because that's not my expertise. But allow me to interest you in several aspects on why I think this is very important. In order for you to support us in the wildlife management and conservation curriculum program, I would like to invite you to consider partnering with us, and especially Dr. Muchai, as adjunct lecturers, visiting lecturers. You can do guest lectures online. You can do a lot of engagements in that aspect. We also would like you to assist us in placement of our students, you know, for attachment and as well as internship at your facilities. When they apply, please just assist us in that. Another aspect is where we are looking for external examiners of our thesis and our undergraduate exams. I think Dr. Muchai, please capture this contact so that we can use them for that purpose. We also have two other things: the research grant proposal writing, where we partner with one another, university, National Geographic Society, National Museums of Kenya, iNaturalists, and all those community of researchers. Let's partner and do research grant proposals. And then finally, we are planning to do a comprehensive review of the wildlife BSc and Masters, perhaps BSc curriculum. And again, Dr. Muchai, I think this is a group that you need to engage. And so I pray for their support. Having said that, allow me then therefore to exit my remarks. I used to have a colleague who had a name Musili, so we'll probably engage to find out which Musili is this. But for now, let's stick to science, and Dr. Muchai, you will help me network with those individuals at a later date. Thank you very much. I may listen, but for now, just allow me to go to the other virtual meeting on leather science. Asanteni, and welcome to Nairobi University.
Okay, good. Thank you very much, Professor, for that very nice opening remarks from the University of Nairobi. Yeah, it's true. We will allow you to at least participate in these other also very important functions. May God bless you. Okay, thank you very much. So we move on now to Paul. Well, Paul Musili. Again, let me quickly.
Good day, everyone. I want to take this opportunity to welcome you to this webinar. My names are Paul Musili, and of the Botany Department in the National Museums of Kenya. National Museums of Kenya is mandated with conducting research on cultural and natural aspects of life through the Department of Research, Repository and Collections. National Museums of Kenya conducts biodiversity research geared to conservation of biodiversity. Kenya is a very species-rich country. Located within the tropics, Kenya is also referred to as hotspot for biodiversity. We know that this diversity is very crucial to the provision of a wide range of ecosystem services that are important, for example, in increasing resilience in food security and nutrition, enhancing livelihoods, environmental health, among others. However, like any other country, Kenya is losing in biodiversity at an alarming rate due to destruction and degradation of natural resources. In the last 50 years, Kenya has lost over 30% of its natural habitat to destruction, degradation, and poor farming practices. Through this, it's believed that 50% of wildlife populations and plant communities have been lost. In the last 30 years, there have been some efforts that have been scaled up to protect and conserve the remaining natural habitats. There has been development of species conservation action plans for a wide range of plants and animals, species diversity. However, very little has been achieved in conservation of fungi, yet fungi are key drivers of the ecosystem. Conservation for fungi, therefore, cannot be possible unless the diversity and ecological data, or rather information associated with these species, is well documented. We at the museums are grateful to National Geographic Society for funding this program, which has brought on board researchers and other stakeholders in the bid to document and monitor microfungi species in Kenyan ecosystems. Through this webinar, I hope we will bring on board discussions around opportunities available to ensure successful data delivery. Botany Department and more so National Museums of Kenya will fully support this initiative, appreciates strides that are being made through this project. It's my hope at the end of this program, there will be powerful conservation strategies for the mushrooms of Kenya that can be initiated. I want to take this opportunity to wish you all the best, to all the participants and organizers of great webinar. Good day to all of you. Thank you.
Thank you very much, Daktari, once again. And now that was very nice. So now we are going to go to opening remarks by Dr. Telessa from the National Geographical Society. Welcome, Dr.
Thank you, and good afternoon. I'm delighted to be here today for this webinar on engaging citizen scientists in macrofungi research and conservation. And I'd like to commend all of the organizers for this important event and to thank all of you for your participation. The National Geographic Society is a global nonprofit organization that uses the power of science, exploration, education, and storytelling to illuminate and protect the wonder of our world. Since 1888, National Geographic has pushed the boundaries of exploration, investing in bold people and transformative ideas, providing more than 14,000 grants for work across all seven continents, and reaching 3 million students each year through education offerings, as well as engaging audiences around the globe through signature experiences, stories, and content. National Geographic has also helped to support many citizen science activities around the world, including this one. We believe that citizen science can play a transformative role in accelerating the collection of data to better understand the patterns, status, and trends of species, ecosystems, people, and cultures. We also see citizen science as an important tool for connecting students and interested citizens to scientists and other experts, and as a powerful tool for learning, capable of inspiring and sparking new passions in those who participate. Mushrooms are incredibly important to ecosystem function and understood in many parts of the world. Through this important citizen science project on the mushrooms of Kenya, you have the opportunity to build your knowledge of mushrooms and your skills in identifying mushrooms, while also accelerating the collection of valuable data to inform mushroom research and conservation in Kenya. I'm very pleased that this project will utilize the citizen science tool iNaturalist, which is one of the world's most popular nature apps and a joint initiative of the California Academy of Sciences and the National Geographic Society. iNaturalist, which you'll hear more about later in the program, allows people around the world to share their observations of biodiversity with scientists, experts, and naturalists who help to identify observed species. More than 77 million observations have now been shared on the iNaturalist platform. The beauty of iNaturalist is that anyone, anywhere can make a critically important discovery. By contributing observations of macrofungi that you observe in nature, and by helping others to identify the macrofungi that they've observed, you have the potential to help discover new species, help scientists better understand the range of species, and much more. The more people who contribute to this platform, the faster science progresses. And in the case of this project, the more that's known about the mushrooms of Kenya, the better they can be conserved. I'm personally very excited to hear today's presentations, and I hope that today's webinar and the one that follows will inspire many of you to become active users of iNaturalist and strong contributors to this important project on the mushrooms of Kenya. Thank you for your attention, and I'm looking forward to the rest of the program. Thank you.
Okay, thank you very much once again. Daktari, here we call you Daktari, doctor in Swahili. So we are very grateful for those opening remarks from our three guest speakers. Thank you very much, and God bless you. Now, because of time, obviously, now we are going to move to our presentations. So we have a number of presentations, 5 of them. So we will start, like I had said before. Again, those of us who are continuing to joining, please feel welcome. So we have Dr. Kilika. Will take around 15 minutes, followed by Dr. Mary Nyawira on the role of citizen scientists in mushrooms. And then after that, we'll have Susan Kafashia, Mushrooms Research Methods for Citizen Scientists. We'll have another presentation from Dr. Carlos. Again, now this one is from iNaturalist, and it will look at iNaturalist tools in mushrooms research. And finally, I will give a presentation on macrofungi conservation strategies. And then after that, we will have our questions and answers. So like we have just said, please, presenters, feel free to put your questions in case you have any as we continue listening in the chat section, and then we'll be able to follow it after the presentations. So right now, I want now to call upon Dr. Paul Kirika. He is going to give us an overview of the fungi research. Overview of the fungi research in Kenya.
Hello, everyone. My name is Paul Kirika from the Botany Department of the East African Herbarium at the National Museums of Kenya. I'll give an overview of the fungal research in Kenya, specifically the research carried out at the East African Herbarium. First, I would want to thank the organizers of this meeting for inviting me to give a presentation. And I will start by giving a brief introduction of what fungi are. And fungi are small filamentous organisms comprised of masses of thread-like hyphae, which constitute the body of the mycelia of the fungus. And they can exist as single-celled, for example, yeast, or as multicellular body comprising of interconnected network of mycelia. They are found everywhere in all ecosystems and in all habitats, including aquatic and both aquatic and terrestrial habitats. And they have some important characteristics. They digest food externally by excreting enzymes into the environment and absorbing the dissolved organic matter back into their cells. So they basically live in their food. And most have cell walls composed of chitin, a substance which is found in the exoskeletons of insects and shells of crabs and lobsters. They also store their food reserves as glycogen and lipids, and not starch as in plants. Genetically, they are distinct, and their DNA indicate that they form an independent kingdom, which is more closely related to the animal kingdom than the plant kingdom. Fungi are heterotrophic and therefore cannot be able to make their own food. So they get their nutrition through various means, including living as saprops, where they break down dead organic matter. And in the process also, they cause a lot of destruction to food, leather, and timber, and therefore causing huge economic losses. Others live as parasites, where they feed on living organisms. And in this process, they also cause diseases. So most of the but pathogens for both plants and animals are parasitic fungi. Others live in symbiotic relationships. For example, the mycorrhiza fungi, which form symbiotic relationships with the green plants, and also the lichen forming fungi, which form symbiotic relationships with algae or cyanobacteria. Others live as predators, where they trap small microorganisms such as amoebas and nematodes. They do so by secreting sticky substances over the surface of their hypha, and therefore causing any animal that touches any portion of the mycelia to stick firmly to the hyphae. And then the hyphae grows and penetrates into the tissues of these animals and release digestive enzymes. Fungi are important ecologically. They sustain life on Earth by decomposing organic matter, breaking it down into simple molecules and releasing carbon, oxygen, and nitrogen, and even phosphorus. Release it back into the soil and to the atmosphere. So they're important in nutrient recycling. Mycorrhizal fungi also provide key benefits to terrestrial ecosystems by enhancing the uptake of nutrients and water by plants, and also are important in the formation of soils, and also they are important in carbon sequestration. They act as sinks for plant carbon. Since time immemorial, man has always used fungi for his needs, the production of foods and beverages. Over 350 species of edible mushrooms are collected world over for food. And some species of fungi are also used in the production of, for example, meat substitutes, in the ripening of cheese and bread making, and also production of food colors. And alcoholic beverages, for example, all the beers, wines, spirits, the liquors like Busa, Moratina, and Kalovu, they are all produced through fermentation by yeast. The uses include enhancing plant growth and in the production of medicines and drugs. For example, penicillin, which is used for the treatment of bacterial infections, and cyclosporine, which is an important drug in organ transplant. Fungi are also used in the production of hallucinogens. In fact, more than 200 species are believed to be hallucinogenic, and majority of which are found in the genus Psilocybin. Other application of fungi to human use include production of enzymes, which are used in the making of washing detergents, processing of leather, and paper manufacturing. Today, fungomycelia is also used in making of substitutes for leather in building, and also in the production of biofuels, where they are used to break down agricultural waste such as maize and sugar straw, which is fermented using then fermented using yeast to produce ethanol. They are also used in bioremediation, where they are known to degrade PCB chemicals in soil and polluted waters. One of the most important components of research is the documentation and preservation of the fungal diversity. Kenya is known to have a rich fungal diversity, and they are studied through collecting, visiting different regions and collecting specimens and data that is associated with those specimens. And as you can see, these are just some of the fungal diversity found in Kenya. And so to study fungi, scientists collect herbarium specimens and data associated with these collections. And these are the vouchers that are used in taxonomic research, in ecological and biochemical, in ecological research and biochemical analysis. And they form reference material for the accurate identification of fungi, while also providing a reliable data on the distribution of taxa. And they are also the core material upon which application of scientific names are based. These are mainly the type specimens. Therefore, give a permanent record of species at a particular time and provide information that are used in the production of fungal checklists, and also information, important information on monitoring. Another area of research is the establishment of fungal cultures, both mushrooms and mycorrhizal fungi, for use in agriculture, forestry, and in the restoration of degraded habitats, and also in promoting mushroom farming. Good quality mushroom spawn is produced for farmers so that they can have a healthy crop, and therefore able to have a good product. The other area of research is the wild edible mushrooms, which this is spearheaded by our colleague, Dr. Joyce Jeffwa, who recently got a grant through NRF. It's out of the realization that mushroom has a great potential to diversify diet and contribute to food and nutritional security in this country, and also increase job opportunities in improve livelihoods. So the project is focused on studying the taxonomic diversity of indigenous edible mushrooms, and also determining the distribution, ecology, and production capacity of these indigenous edible mushrooms, while also evaluating the different methods of preserving the genetic materials, and also trying to devise appropriate ex situ and in situ conservation approaches for the indigenous edible mushrooms, and also determining their safety through carrying out nutritional and toxicity analysis. The other area of research is the study of mycorrhizal fungi, which are important in the maintenance of plant species communities, habitats, and systems. And these involve the study of vascular mycorrhizal fungi and ectomycorrhiza. And it's important to point out that in the conservation of rare and endemic plant species, the study of the associated mycorrhizal fungi is of utmost importance. To conclude my presentation, I would say that the fungal diversity in Kenya is not very well documented, and data on fungi for most areas is lacking. And this gives opportunities for fungal fungal research in taxonomy, evolution, ecology, physiology, genetics, and even chemistry. And that is the end of my presentation. Thank you very much, and would welcome any questions or any comments. Thank you.
Okay, thank you very much, Dr. Paul Kirika. So I'm requesting participants, if you have questions or comments, that you put them in the chat section. You just write them down there so that we are going to look at them after the other presentation. So please feel free to put your questions and your comments in the chat section. Yes. Now, we are moving on now to our next presentation, which will come from Dr. Mary Nyawira, who is from the National Museums of Kenya. She's going to present on the role of citizen scientists in mushroom research. So welcome, Daktari, for that presentation.
Our guests and all participants, my name is Samire Nyawira Moshane, a research scientist at the National Museums of Kenya. I am here to give a talk on the role of citizen scientists in mushroom research and conservation. Welcome. Before we look at the role of citizen scientists in mushroom research and conservation, we will first see what is citizen science. Citizen science is scientific research conducted in whole or conducted in part by non-professional scientists. These non-professional scientists may even include the amateurs. In other words, we can define citizen science as an active involvement of general public in correcting or active involvement of general public in correcting and analyzing of scientific data. These non-professional scientists are usually volunteers with a passion in an area of study. They can be local conservationists, they can be tour guide, they can be scientists, they can be students, farmers, or any other person interested in contributing to science. These non-professional scientists are the ones we are calling citizen scientists. Why should we use citizen science in research and conservation? First reason why we should use citizen science in research and conservation is that it is a very cheap way of acquiring scientific data. Citizen science program utilizes people on the ground. It thus reduces the cost of accommodation. It also reduces the cost of transportation, making it a very cheap way of acquiring scientific data. The second reason why we should utilize citizen science program in research is because one can cover large area within a very short time. When using citizen science program, one is able to recruit a wide range of people from different locations, making it a very fast way of acquiring scientific data. Citizen science program also enables scientists create local awareness on the role of science in development. For us in biodiversity research and conservation, for example, when we use citizen science program, we are able to create awareness on the role of biodiversity in our ecosystem. We are also able to create awareness on why the local community should support biodiversity conservation programs. When we use the citizen science program, we are also able to enlighten the citizen scientists, or we are able to enlighten the local communities. And in doing this, we are able to improve their scientific literacy. We are also able to improve their capacity. And now these people can contribute to science. They can also contribute to conservation significantly. When we have now enlightened local community, the adoption rates of scientific results as well as conservation technology is increased drastically. This is because we do not have now to convince these people to know the importance of a given scientific result, because they are already aware of the importance of this result in their ecosystem. They are also aware of the consequences if these results are not implemented. Although citizen science programs have been used widely in research and mapping of various taxa like birds, mammals, plants, the programs are rarely used in fungal research. That's why we came in early 2019. We initiated a Mushroom of Kenya project with the aim of understanding the role of citizen science in mushroom research and conservation. This work was motivated by lack of macrofungi diversity data. It was also motivated by lack of macrofungi ecological data in our Kenyan ecosystem. And due to lack of this data, there has been no effective management and conservation strategies that have been developed for these species. The consequences of this is that the fungi has remained overlooked in major conservation initiatives, and yet we know that the macrofungi diversity or the mushroom diversity, it is very key in sustainability of our ecosystems. Before we continue, I want to give a brief description of what macrofungi are in general. Fungi are living organisms. They are not plants. They are not animals. Fungi are classified in their own kingdom, the kingdom fungi. In this kingdom, we have two categories of fungi. We have the microfungi. These are fungi that we cannot see with our naked eyes. We can only see them clearly using a microscope. The second category of fungi is called the macrofungi. These are fungi with a visible fruiting body, or they are fungi we can see with the naked eyes. There are different categories of macrofungi. We have the mushrooms, we have the puffball, we have the bracket fungi. However, most of us know the mushrooms. Mushrooms are usually characterized by having a cap. A cap is the top part of the mushroom. It can be smooth, it can be scaly, or it can have scales. It can be hairy. Beneath the cap, we have the spore-producing tissues. These can be gills or they can be pores. Beneath the gills, we have the stipe or the stem of the mushroom. The stipe supports the cap and connects the cap with the mycelium. Beneath the stipe, we have the mycelium. These are thread-like roots. They attaches the mushroom to the substrate or the media where the mushroom is feeding from. In general, a mushroom is usually made of a dense mass of mycelium, or what we are calling the thread-like roots. Why do we study mushrooms? We study mushrooms because they are key in sustainability of our ecosystem. In their natural condition, most of the mushrooms are decomposers. They release nutrients from the dead organic material back into the soils, and in doing this, they make nutrients available for the living plant community. As a result, they are able to maintain a stable plant community. Secondly, some mushroom species form mutualistic relationship with the plant. From this relationship, mushroom species help the plant take up nutrients and water from the soil. They also help in control of root-related diseases, and in doing this, they help maintain a productive and healthy plant community in our ecosystems. Another reason why we study mushroom is because they are key in climate change mitigation. The density hyphae of these mushrooms in the soil help in development of micro and macro aggregates. We know these aggregates are very key in the storage of carbon in our soils. We also study mushroom because they are very key in cleaning of our polluted environment. Mushrooms have ability to break down the chemical compounds found in major pollutants in our environment, and in doing this, they help maintain a clean environment. We also study mushroom because some of these mushrooms are parasitic to our plants. They are also parasitic to our crops, and they cause significant economic losses in our crop and tree production. So we have to study these mushrooms or these fungi so that we can be able to manage them and maintain productive ecosystem and productive agriculture. Mushrooms are also very key in improving local livelihood. Mushroom cultivation enterprises, for example, they provide both food and income to the farmers, and in doing so, the livelihood of these farmers is usually uplifted. After short descriptions of what mushrooms are and their importance, we are coming back again to see why we should engage citizen scientists in mushroom research and conservation. Mushroom is usually a very unique taxa. It is unique in such a way that it has a very short lifespan, a lifespan of 1 to 20 days. It is therefore very easy to miss a species if you come for correction or if you come for sampling when its lifespan has ended. Mushrooms are also unique in such a way that they are affected by season. Mushrooms mostly occur during the rainy season, and not all of them will appear at the same time during the rainy season. There are those which appear at the beginning of the season, there are those who appear at the end of the season, and there are those which will appear at the middle of the season. This means that the short survey usually carried by scientists will always underestimate the mushroom diversity in our ecosystems. That's why now we need to engage the citizen scientists. These are the people on the ground. They are the ones who can document these species when they are sprouting. They are the ones who can document these species across the season, and they are the ones who can now document these species before their lifespan has ended. We are also using citizen science program to enable us create local awareness on the importance of mushroom community in our ecosystem. And we know when we do this, we are going to improve the scientific literacy of our local communities, and we are going to enable them contribute significantly to mushroom research and their conservation. We are also using citizen science program to help us generate macrofungi diversity data, a data that can be used in development of management and conservation strategies for these species. We also want to gain data that can help in expanding the mushroom industry in Kenya. We also want to gain data that can be used in developing effective afforestation program, especially for tree species that form mutualistic relationship with some of these fungi species. At the same time, we want to generate data that can be used to develop a field guide that can be used by both the scientists and the citizen scientists in describing and identification of mushroom or macrofungi species in Kenya. As I said before, in January 2019, we began a pilot study to test the effectiveness of citizen science program and iNaturalist data collection tool in mushroom or macrofungi data collection. In the next lines, I will show the potential citizen science program and iNaturalist data collection tool have in mushroom or macrofungi data collection. In this pilot study is currently being done in Aberdare Forest. In this pilot study, we first developed a mushroom monitoring program. In this program, citizen scientists are collecting mushroom specimens from already established permanent plots. This monitoring program is being tested in 2 sites in Aberdare Forest. The first site is in southern Aberdare, that is Greater Forest. In this site, we have recruited 7 citizen scientists. This far, they have managed to collect over 3,000 mushroom specimens. These are translating to over 500 mushroom species. In the other site, which is in West Aberdare, the Greater Forest, we have also recruited 7 citizen scientists. This far, they have managed to collect over 1,000 mushroom specimens, and these specimens are translating to over 200 mushroom species. We also developed a Mushroom of Kenya project in the iNaturalist. For those who have not come across the iNaturalist is a free online platform It helps in recording and sharing of biodiversity observation using photograph. The use of this program or platform will be discussed later in details in this webinar. When we developed this iNaturalist Mushroom of Kenya project, we developed it to capture mushroom data collected all over Kenya. We wanted to capture all mushroom data collected by other passionate citizen scientists. This far, the project has received over 2,500 mushroom specimens. These are translating to over 400 mushroom species. From this data collected by citizen scientists, using iNaturalist data collection tool, we are now able to see how mushroom diversity is distributed across Kenya. From this map, we can see how mushrooms are distributed all over Kenya. We can see mushroom observation in all regions in Kenya, with the exception to northern eastern Kenya. When we translate this data into a graph, we can see very high mushroom density in central Kenya compared to other regions. This is understandable because this is where we have a detailed monitoring program in Aberdare Forest. Using this data, collected now by the citizen scientists, we can now see how mushrooms are distributed across different counties in Kenya. In this slide, we show how mushrooms are distributed across the Kiambu County. Using this data still, we can also see how different macrofungus species are distributed across Kenya. From this map, we can see how Agarika species are distributed across Kenya. It is very interesting to see Agarika species even from the northern part of Kenya. From this work, we have discovered a wide range of macrofungus species. The species have ranged from mushroom, toadstools, puffball, stickenhorns, polypons, asters, and the like. We have also discovered a wide range of edible and medicinal species. Some of these species are being captured to preserve them for future research and for future use. From this work, it is clear that many of these species we discovered would have taken long before being discovered or would have taken long before being documented. We therefore thank the Citizen Science Program and also the citizen scientists for this great progress. In conclusion, I can say that Citizen Science Program and the iNaturalist data collection tools have potential in revealing macrofungus species in our Kenyan ecosystem. From the Mushroom of Kenya project under the iNaturalist, we have discovered over 400 mushroom species. These are from over 2,000 observations. I cannot confirm this is the highest number of macrofungus species ever discovered in our Kenyan ecosystem. From the detailed monitoring program in Aberdare Forest, we have discovered over 500 mushroom species from over 3,000 observations. I can also confirm that this is the highest number of macrofungus species documented in our Kenyan ecosystems. We are therefore calling each one of us to join this citizen science program. The data from your observation and my observation will be very key in developing management and conservation program or technologies for macrofungus species in Kenya. The data will also be very key in expanding mushroom industry in Kenya. This data we are collecting will also be very key in supporting a forestation program in Kenya, especially for the tree species that form mutualistic relationship with some of these mushroom species. This data will also help us develop a field guide that can help in descriptions and identification of mushroom from our Kenyan ecosystem. Through this program, we are also going to gain knowledge in mushroom taxonomy. This knowledge will help us contribute significantly to science. We can also get engaged in identification of mushroom species submitted to iNaturalist online repository. Let us therefore join hands in making our mushroom diversity known to enable their sustainable management for healthy and productive ecosystem in Kenya. As I conclude, I wish to thank the iNaturalist team and the California Academy of Science and the National Geographical Society for supporting this webinar and for supporting this work. I also wish to thank the staff of National Museum of Kenya, Nairobi University, and all Kenivo staff for supporting this work. I also wish to thank all citizen scientists who have contributed their time and resources to make sure mushroom research does not lag behind. Let us keep up the good work. I also wish to thank all of us for joining and listening. To all of us, thank you, and let us keep mushroom research ongoing.
Thank you very much, Dr. Mary, for that nice presentation. So I hope participants, those of us who are continuing to join in, please feel welcome. If you have questions for any of our presenters, just write the question or the comment on the chat section so that we are going to answer them or deal with them after the presentations. So right now, ladies and gentlemen, if you allow me, we are going to move on to another presentation. This presentation is by Susan Kafashia of the National Museums of Kenya. She's going to deal with mushroom research methods for citizen scientists. So Susan.
My name's Susan Kabashia. I'm a research scientist at the National Museums of Kenya. And this evening, I'll be presenting on mushrooms. And this is on the collection methods that are basically used by the citizen scientists. Two types of methods are used. And they're mainly manual collection and the digital collection. And on the manual collection, this is where the citizen collects data in the field or collects samples in the field and submits them to a research scientist, or submits the observations and the notes to the research institution. And the other method of collection uses the online data collection tools, where the observations and the notes are submitted online. And there are requirements for both the methods, and this is whereby the collector should have basic training on how to collect mushrooms in the field, and also to have the understanding of how the mushrooms look like. Secondly, he should be aware on the collection seasons, whereby there are months that are suitable for collecting data or materials, and this is mostly during the rainy months or seasons. The other thing is that he or she should have the knowledge on handling different types of mushrooms. This is because mushrooms are of different forms, and at the same time, it's also good because he or she can take a precaution, and especially for those mushrooms that are known to be poisonous or to be toxic. The other aspect is having the knowledge on what information to document or what information to accompany the specimens, or rather the mushrooms that he or she has collected in the field. And it's very, very important to note that not all mushrooms are edible, and there's those mushrooms that are poisonous or toxic, and therefore, unless one is familiar with the edibility of the mushrooms, then it is important to take note so that he or she is not poisoned. The other aspect on digital collection, and it's a requirement, is having acquiring very good photographic images which are downloaded online using the data collection tools, where in this case or in this project, we recommend the use of the iNaturalist. And this is where the photos are uploaded together with the notes on this platform, which is an online repository where now scientists come and they can be able to identify the specimens, or further, or even for their own research. And this helps so that in case some species are not known within the country, or they are not people that are familiar, especially those who are collected with the specimen identity, then they can be identified by other research scientists around the globe, possibly at genus level, at species level, or even at family level. And before collecting samples in the field, there are also requirements. And one of the requirements is to acquire permits and permissions. And this is because mushrooms are found occurring in different entities, which are some are government protected areas, others are private farms, and it's important to have permission to access such places. When it comes to the collection, it's also important to have equipment and also to be well adorned for the field work. And this is because as far as the mushrooms are concerned, they grow in different habitats on different substrates. So there are tools that are important actually in obtaining the collections. Some require uprooting, some requires cutting off the substrate. It's also good to take images, and that is why we need good phones and a good phone with a good camera. It's also good also to have a camera if need be, and also, and that is not when need be. It's an important gadget, but as well, the phone can also play the part of a camera. And this we are going to see as we move on in our next slides. For the personal safety, it's important to have protective clothing. It's good to have gumboots. It's a rainy season, so it's important to have a raincoat. It's good to have a first aid kit in case of any form of an accident, sometimes they are minor. It's important to have these gadgets in place. Then it's important for the collector to have a field guide. It helps, it plays a role in identifying with the different forms in nature, and also in very basic description or identification of mushrooms that are growing in the wild. And it's also important to have the knowledge on where the mushrooms are growing. They grow in different places. They grow in different locations. So we find some in the forested areas, some in plantation forests, and also their feeding also is different. Like for the saprophytic fungi, they found growing on wood. There are some that grow in association with specific trees, or rather, there's some trees that cannot grow without association with mushrooms. So it's important when documenting information that this information is captured. Some will be growing on living trees, and this is because of their parasitic nature. And then there's a process now of collecting mushrooms, and it is very key for photos to be taken and good photographic images. And this is of the habitat where the collection is being done, because the site ranges, the grassland, plantation forest, indigenous forest. This is key when identifying and also when associating some mushrooms in the habitat where they grow. And also on the specimen, it's good to get good images showing all the views of the specimen. It will be on the upper part of the cup, under the cup, the style. Every detail of that specimen should be captured in a photograph. Then also, it's also important to take the specimen with the scale where a ruler is placed, and it can show the dimensions of a given specimen. Then when it comes to the label, it's important to place the label close to the specimen, because this reveals a lot. It reveals where the specimen was collected, the number and the date, and this helps when placing a specimen with the right data. And consideration should be given when collecting samples, that they should be of good quality. This is specimens that have not decayed, the specimens that are not very mature. The other aspect is on the developmental stages. It's good when collecting the samples to collect all the specimens with their different developmental stages. And also, it's also important to understand that because of the different sizes or different nature of growth, how to collect. And for the very small mushrooms, they can be uprooted very well, making sure that all the details about that specimen have been captured. Then ones that are growing on wood, they can be sliced well, so that a whole specimen is obtained. Then for the soil dwelling, they can be uprooted well using a scooper, making sure that the base, or rather the site, is not damaged. It is uprooted completely. And when it comes to the sample size, you find that because of the different sizes, the quantities also matters, whereby for the very small sized mushrooms, 15 to 20 specimens will be okay to collect. For the medium, 5 to 10. For the large ones, at least 2 specimens will be enough. Then this is now the information that should accompany the specimens. And this is normally in form of a sheet. And this sheet contains the collector's details, which includes the collector's name, the field number that the collector has assigned to his sample or her sample, the site details, is which area was this mushroom collected, what was the land use, is it indigenous plantation, is it a farmland, was it growing on soil, on litter, on stumps, and what were the associated plant species. Then the other aspect that should be considered is the specimen itself, the crate. What color is it? What texture? Does it have odors when smelled? Does it change? Does it have color changes when bruised, or rather when it is cut? It is important to capture such details. Then labeling is key, as we said, and this is because the label carries important information, and it should be tied to the specimen. And the label or the label and the specimen should be placed in a paper We normally use the greaseproof papers, and then the specimen is placed in the basket, ready for transportation to the research institution. The collection should be submitted, if possible, the same day. And if it is not possible, then they should be dried or stored in the fridge. Drying should be at 40 to 45 degrees centigrade. And if kept in the fridge, it should not be more than 2 days. Before drying, it's important to take a spore print. This is whereby the cap of a mushroom is cut, is placed on a black, white, or green paper. It usually forms overnight. It usually forms overnight or after a few hours. And it is placed very well in a Ziploc paper and stored together with the other specimens. Then once the specimens have been submitted to a research institution, then further identification takes place. The specimens are further described, they are dried, they are stored in airtight containers, and after that, they are curated for future research. For the submission of observation online, then after the collections have been made, they have been submitted, then photos can be uploaded together with the notes using the recommended online data collection tools, and in this case is the iNaturalist. And the images should be of good quality, and the site and the specimen details should be conclusive. I want to acknowledge the National Geographic for funding this work. I want to acknowledge the National Museums of Kenya and the University of Nairobi for the research that has been carried out in this project, and also Kenvo for helping us, and for the team that you have worked with in this project. And finally, I want to say thank you to all for listening.
Thank you very much. Thank you very much, Susan, for that very nice piece of presentation. So now, I still request participants, if you have questions or comments for that presenter, you just put that question or comment in the chat section, and then we are going to look at it after the next two presentations. So right now, we will continue to with our presentations, and the next presentation now will come from Dr. Carlos, and he's going to look at iNaturalist tools in mushroom research. Welcome, Dr.
Thank you so much. Hello, everyone. Thank you for this great opportunity. I'm currently living in Northeast Mexico from across the world, so it's really, really great to be here with you today and share a little bit about this digital tool that iNaturalist is right now. So I'm going to start my sharing screen. I hope this works. Okay. Okay, so I hope that you are seeing now my screen, and I'm going to talk a little bit about how iNaturalist it's one of the greatest tools that researchers and people across the planet can have to actually collaborate in the knowledge and conservation of biodiversity in every single part of the world. So, okay, what is iNaturalist? We have heard a little bit about, okay, iNaturalist is this online tool, but there's no single definition of what iNaturalist is, because it's a really flexible tool that can be used in many different ways. So iNaturalist is really a lot of things at one time. We can say that in a rough definition, iNaturalist is pretty much like a social network. I mean, social networks are all over the world, and we use them in different ways. But iNaturalist is designed not for scientists, but for people, for community, for general public. And it's a way to reconnect people with nature, with biodiversity. Of course, this comes with an extra bonus that it's gathering information on biodiversity. And one thing that it must be said is iNaturalist is not to replace collecting specimens for museums or herbariums or collections from any kind. iNaturalist is just a way to complement information that scientists, well, will be having some troubles to gather by their own. Remember that we are facing a massive extinction in many different groups, and we need all the help that we can get to fill the gaps of information on biodiversity. So iNaturalist came just in the right time of humanity to help us document biodiversity. And believe me when I tell you, iNaturalist is a global effort, and it's all over the world, and many countries are joining in as a national node to promote among their citizens the use of this kind of tools and to empower local communities in the knowledge and documentations of biodiversity. And it has been evolving in the few years. iNaturalist started on 2013 as a master's degree project from a couple of students in California. Right now, iNaturalist is a joint initiative between the California Academy of Science and the National Geographic Society. This is important to say, because it really brings weight to the fact that people are collaborating all over the world. So you're now seeing information from last year, when iNaturalist managed to get 300,000 species already being documented in this platform. We might say that, okay, 300,000 species is not that much in terms of the whole biodiversity in the planet. But believe me, this is an unprecedented numbers in terms of people gathering together to document biodiversity. And we have, okay, some of the most charismatic groups like vertebrates, like reptiles, birds, and mammals have already a good number of species already documented on iNaturalist. But let me try to, okay. I hope, oh yeah, this works. So let me close this. No, it's, sorry. Let me try again. We're going to focus on particularly this group, fungi. You can see here that this little corner here says that last year, iNaturalist has only fungi. It was represented by almost 30% of all the documented species. This might seem like a really small number, but for this group, especially this group, that it's not easy to work with, because sometimes you will need a lot of information to have like specific identification for a species. This is actually a great accomplishment for iNaturalist to have at this point. This is how iNaturalist looks today. Well, a couple of days ago, I took this screenshot a couple of days ago, but it really shows the true global reach of iNaturalist as a worldwide community. You can see here that we have, well, 1.8 million users in the whole planet. This is really an incredible numbers in terms of most of these users are not scientists. It's just people who is interested and likes to get in touch with nature. And it's really helping scientists across the globe to have millions of observations. And when we talk about millions of observations, here is this huge number, 76, almost 77 million observations. And if we go through species, we can see that there's more than 300,000 species now. We are about to reach 350,000 species today. And another really important part of this online community is identifiers, people who is willing to help other users to get identifications of their observations when they uploaded. And 200,000 identifiers in this global community is a huge number, because among these identifiers are top world experts that are helping other people to identify these observations. This is what makes iNaturalist one of the leading citizen science platform in the whole planet, collaboration and community. Okay, so we have millions of observations, but specifically for fungi, there is a growing interest, and we have what it might seem as a low percentage. I mean, 5% of all these millions of observations are just fungi. This is roughly 4.2 millions of observations, and we have so far 15,000 species. But again, we have a huge number of observers. This is what really important is, because it will make growth these other two numbers in the near future. And again, identifiers, almost more than 41,000 identifiers helping people to identify. This is for the whole planet. But what if we're going to be narrowing down? This is what we have for the whole continent in Africa. 50,000 observations, 1,400 species. And again, we have a lot of people doing observations which will really help biodiversity understanding to grow in the next years. This is an untapped potential for people and researchers to start growing collaborations in the near future. Of course, so far, some of the most conspicuous and visible observations are being made are microfungi. And this is the top species that are being observed in the whole continent. I'm sure most of you will easily recognize some of these because these are really easy to find or really common species. Some of them are quite charismatic, like this anemone stinkhorn fungus. People are taking photographs, so you will be having a lot of information to interpret and track for this particular species. We can also take a quick look at the major groups of the microfungi, like Basidiomycota, where we can find some of the more common macrofungi. This is that we have only 959 species documented, but again, a lot of observers here. This is what we should be really putting an effort to promote. And as a scientist, we will have the opportunity to work with all this information as you have been doing already in the last few years. And again, another really important area that has a great area of opportunity is lichens, which are located on the Ascomycete area, or at least this is where this is being classified here in iNaturalist. And I'm not going to discuss like taxonomic details on iNaturalist, because iNaturalist has this ability to try to put together a taxonomy or classification that fits most of the world. I mean, you may sometimes not agree completely with taxonomy here, but this is not the place for a discussion on taxonomy. This is a place to get collaboration on biodiversity documentation. So really, I really want to say that this group of microfungi, lichens, has a huge potential for knowledge in this area. And how does Kenya stands in terms of biodiversity documentation using iNaturalist? This is a whole picture of the country and how it looks today in terms of observations, user, and species. You have a very nice number of observations, almost 7,000-8,000 observations here in the whole country, with roughly more than 6,000 species already been documented for the whole country. You have also a good number, you have more identifiers than users. We have to make sure that in the near future, you will be promoting the use of iNaturalist so you can grow the local number of observers and communities and people from every part of the country start using this so they can empower themselves as a community to make observations on iNaturalist. But if we go down to the fungi, then again, we have a little number so far already documented on iNaturalist. We just are reaching 300 species already been documented on iNaturalist when more than 2,000 observations, and we have 58 observers. This gives us a really huge room for growth, for refining observations and how we are using and how we will be using iNaturalist in the futures to build up on knowledge for research and local conservation. And here in this particular slide, this is what we have as research grade observations. This is observations that have already been refined, reviewed by other iNaturalist users. And here again, I'm going back. Oops, sorry. We're going back to this slide, 330 species and 39 species on research grade. We're going to see in the future session how researchers can help other users to actually make this number grow, because there's already a great amount of fungi observations that have been correctly identified, but has not been verified by researchers on this platform. And here is where we can make a huge difference for the local communities to start really appreciating and documenting more precisely biodiversity on macrofungi. This is, well, how we can see on really macrofungi, Basidiomycota. These are the 295 species that are already present here, and there are verifiable observations. We might have another number of species that are not yet verifiable because they will lack some information. But again, these are information that can be curated, that can be reviewed, and can be corrected or edited to get more accurate and rise the number of species already documented here on iNaturalist. This is, well, the number of Ascomycota where lichens are present. You can see that you have only 5 so far, 34 species with less than 200 observations. Again, this is an untapped research area that some of you, especially students, might be interested in reaching for research. And what happened when a student or people from among the community are interested in citizen science on this particular group? And I want to share this particular example of this fellow student from here, my city in Mexico. His name is Alejandro Huereca, and he was an undergraduate student back on 2017. He was just a regular student who actually was working with reptiles. He was an herpetologist who liked snakes, who liked lizards. But at some point in his mind, he just said, okay, what about fungi? What about lichens? And then he joined it on October from 2017 on iNaturalist. And he started to pour all his information on lichens. And I have to say that we don't have local lichen or fungi experts. This student started to reach out for information on his own. And then he started to pour information on iNaturalist of his findings. And you can see, this is my state here in Northeast Mexico. I live close to the border to the United States. And we have a very diverse ecosystem area from really desert areas to montane and humid and tropical forest in the mountains. But when he started to add all the information on lichens to iNaturalist, you will be seeing, okay, we have like just 126 species on 2017, and then 2060 and 64 species in the next year. And observers also started to grow. We have 48 observers the first year after he started. Then we have the double of that, 1,011 observers. And observations also started to grow in a really high rate. This is 2019, before the pandemic got us. And we already have 376 species of lichens and Ascomycota species in the state. This is just the state where I live. And again, observers started to grow as they saw that these species were adding, and we were raising awareness about this particular group of fungi. What happened in 2008, even as the pandemic were hitting us, for this particular group of fungi, we reached more than 4,000 observations. We reached 400 species and more than 100 species, and observers still growing, even surpassing the number of identifiers, even if this group is not easy to identify just by photographs. Alejandro and other fellow researchers were starting to add detailed photographs on how to make better identifications of this particular group. They were even adding observations of microscopic photographs showing the proper details to get correct identifications. And even during this year, observations continued to grow on this particular group. iNaturalist has a lot of unique observations, from the most common of the species that has hundreds of observations, like one of the first slides that we saw, to truly unique observations like this poison fire coral mushroom that was observed in Japan. These are like unique observations, only really like true jewels that are found. And there are a lot of really amazing discoveries that are being made because people are just sharing these observations on iNaturalist. Some of them are like pre-observations, like great photographic, but some of this will actually lead to scientific discoveries. And I'm not going to talk about that, something that happened in some other part of the world. This species that is now called Alexoria sierramadrensis, I photographed this particular lichen back in 2008. It was just like a and it was photographed in a natural reserve within my city. My city has like 4 million people living on it. We have air pollution, we have water pollutions, we have biodiversity loss. We had a lot of environmental problems, but we have some natural reserves already within the city. You can see here, this little dot here. We were able to document this. And when people saw this on iNaturalist, said, this is interesting, we should collect some samples. And again, this is part of the process of researching fungi. I mean, you cannot just identify some of the species just by photographs. So you can take a sample, you can go to the lab and start the science process to get identification on this. And this is an observation of the same species, but you can see that this was taken far away from the first observations. So we were able to document this species from a different mountain range within the city. You can see that the city is just, well, like most cities are, just a lot of concrete and buildings in there, but then you can see some natural areas around. And you can see here that photographs from the spores are added here into this iNaturalist observation. You can see also, well, it's not easy to see, but there's also chemical reactions photographs to show how it behaves. We know that some fungi will need like chemical reaction to actually have like a proper, better identifications. But after a few years of research on these particular specimens, as researchers, we are really interested in publishing papers. And this is one of the outcomes of these observations of iNaturalist. This was actually described as a new species for the country. And look at this date. It was just published on January 20 from this year. And here's one of the authors, Alejandro Huereca, this undergrad student. He's now doing his master's degrees in Canada. But this was like a non-precedent discovery, because this particular lichen was nothing like it was known for this particular group of lichens, and not for this particular group of on this genus like Alixoria. It was like a really, really fantastic discovery. The information of iNaturalist has the potential to describe new species, or to track, and this is another paper that was published using iNaturalist to track ectoparasitic fungi in the US. And you can see in this table, this is just an example on how much information can be gathered from iNaturalist and comparing to other sources of citizen science information. This is just to show how much information can be obtained from this citizen science. And of course, there's even more detailed studies that you can actually track fungal diseases from photographs already on iNaturalist. This is a paper that has been already been published when they were looking for fungi on the anthers of the flowers. You can see this is a healthy flower without infection disease. And then this is an iNaturalist photograph with infected anthers. And this is an herbarium voucher with the disease on the anthers of the flower. And believe it or not, they look for 200 years of information on museums and herbariums. And they actually obtained the same amount of observations just from 20 years of iNaturalist observations. They have the same information than in 20 years than the information on 200 years of herbarium or museum specimens. So these are published papers that have already been using the information that is already on iNaturalist. And this is the example that I wanted to show before we actually, in the future sessions, we're going to have another session for actual training on iNaturalist, how to actually upload, identifier, and really make this community grow. So this is first showing a little bit on how much iNaturalist has been growing. And believe it or not, people from across the planet are really taking interest in how iNaturalist is working. This is a workshop that was presented a couple of weeks ago on the International Association for Lichenology to show our researchers how this has been growing, how this has been used, and of course, to show researchers how to properly use iNaturalist. Sometimes we have little time available, but I think this is a really unique time in history where researchers are taking advantage on using the power of citizen science as a tool to actually have more and more information. And I know that some of you or most of you are interested in mushrooms, but iNaturalist is designed to work with every single species in the planet and different arrays of geographical space. We have been seeing how the world is doing, how the African continent is doing, how the country is doing. But I'm showing you here right now how my house is doing. You can start using iNaturalist into your own house, your own local park. And this is really important for us to know, because then we can tell this to the community, and the community can just start documenting biodiversity in their houses. Of course, there are a lot of new species that are being found across iNaturalist. This is not just for mushrooms. There are flies, moths are being that are seeing found. And one of the examples I really like to share with all the community every time that I have is experiences that I've been having, like this little tree cricket that we found just outside my house. It was described as a new species because iNaturalist gave us the opportunity to collaborate with other experts on taxonomic groups like these tree crickets, which I am no expert with, but I was able to collaborate. And this gives people and scientists the really unique opportunity to collaborate with each other for the advancements of science and the conservations of biodiversity. Again, I really want to thank all of you for having this time here. I mean, we are more than 40 people gathered together to talk a little bit about biodiversity and how we can improve the way we understand biodiversity. This is just a picture of how much iNaturalist has been growing in the terms of numbers of observations. This particular gap you will see here, this is what we need to fill in the next few months, in the next few years to come, to actually preserve biodiversity and get a better knowledge. So really, I'm really happy and for this opportunity. I want to thank you, all of you, for being here and for putting this together. And I'm sure we will be seeing in the next sessions. Thank you so much for your time and your patience.
Oh, good thing, good. Thank you very much, Dr. Carlos, for that nice presentation about the iNaturalist tool. And I'm sure, like he's saying, in the next webinar series, he's going to go further and to show us how now we can use the tool. Thank you very much. Now, we are going now to the next presentation, and that is now myself, quickly. And I am Mushane Mushai from the University of Nairobi. And I'm going to share a presentation on macrofungi conservation strategies. Welcome. Distinguished guests and all participants, my name is Dr. Mushane Muchai. I work for the University of Nairobi, Department of Clinical Studies, Wildlife Section. I am here to share with you a presentation on conservation of macrofungi diversity. I will highlight strategies that can be used by citizen scientists in management and conservation of macrofungi species. Welcome. So the first question to ask ourselves is, what is biodiversity? In simple terms, biodiversity is the variety and differences among living organisms at species level, genetic and ecosystem levels, and also the ecological complexities of which they are a part. Therefore, macrofungi diversity is the variety of the macrofungi species at the 3 levels, and the ecological complexities, obviously, how they interact with the environment and the ecosystem. So another question to ask ourselves is, what is macrofungi diversity conservation? So macrofungi diversity conservation is the practice of protecting and preserving the wealth and the variety of the macrofungi species, and also preserving their habitats, their ecosystems, and also not forgetting the interrelationship between these macrofungi species and the environment in order to keep the ecosystem healthy and functioning. Why should we conserve macrofungi diversity? In addition to decomposing waste, macrofungi provide food, medicines, income, and livelihoods for both urban and rural communities. Yet, despite their critical importance, macrofungi species continues to be under threat. Even as we talk right now, macrofungi species are increasingly vulnerable to the impact of land use, land degradation, ecosystem disturbances, logging and felling of trees, and ecosystem fragmentation, among other vulnerability factors such as climate change. Many biodiversity populations, including mushrooms, are declining due to the loss or reduction of natural habitats in particular. Many farmers have been progressively moving towards cultivation and agriculture. This is drastically changing natural pristine areas. Even the remaining natural habitats are subjected to varying degradation and disturbances, further narrowing the survival of biodiversity and also mushroom biodiversity. Habitat loss and fragmentation are indeed a major causes of macrofungi species extinction. Any activity that intensively removes the woody substrates on which fungi species grow, any activity that will remove the forest florita, or that will remove the shrub host with which the species is associated, or any activity that might remove or lead to destruction of the fungal organisms, or that might lead to the removal of the host species, or significantly modify the microclimate at the macrofungi species site will negatively affect the macrofungi species. Substitution of indigenous species by monocultures, or for example, pine, cyperus, eucalyptus plantations, and other similar species is also causing a major shift in the composition of wood inhabiting fungi. Macrofungi are also vulnerable to unsustainable utilization. All these threat factors are worsened by climate change. Let us look at macrofungi conservation objectives. As citizen scientists, we need first and foremost to come up with an objective of understanding and documenting macrofungi diversity at all various levels, all the way from the national level, regional, and even globally. In order for us to have a clear objective of conserving our microfungi diversity, because we need to know where and which and what are these macrofungi species. The other very important objective that we are also going to look at is using macrofungi diversity sustainably, because if we do not use them sustainably, then they will continue declining at an alarming rate, and they will not be able to survive. Another objective is policy advocacy, promoting education and awareness, especially about macrofungi diversity, and lastly, making sure that we can be able to build up capacity, especially for our key stakeholders for microfungi conservation. But what are the conservation strategies for macrofungi species? We have two basic approaches to macrofungi conservation, and these include, first and foremost, in situ conservation, which I'm going to explain later, and also ex situ conservation, again, which we are going to look and see later. In situ conservation means conservation of species in its natural ecosystem or semi-natural ecosystem. Some of these natural and semi-natural ecosystems include forest reserves, national parks, conservancies, road reserves, sanctuaries, biosphere reserves, ranches, and all sites that are designated for active long-term conservation of species. So let us look at some of the macrofungi conservation tools that are available for our citizen scientists. So the first important one is to make sure that we are able to increase the amount of coarse dead wood that is left in the forests to decay, or in the grassland, or in the various habitats. So, but in most cases, this is very important for the wood inhabiting fungi. So we should make sure that we allow enough coarse dead wood to be left in the forest to decay. The other one is the retention of trees in managed forest. So trees, or the tree group, should be let to grow to maximum age, and again to be allowed to undergo subsequent decay right in the forest without being corrected. In the grassland species will benefit a lot if we allow continued grazing, that is moderate grazing, and absence of fertilization in old grassland. There are those areas that have reduced nitrogen emission, especially in areas with predominantly nutrient-poor soils. Habitats like grassland, for example. So again, for this kind of species that are associated with the nitrogen poor soils, then we should at least try to make sure that there is reduced nitrogen emission. So we have said that species action plan is very key in conservation of macrofungi. So let us look at some of these species approaches. So the first thing you must do is to make sure that you can be able to describe, or the description of the habitat for each of the species in concern, or that you are concerned with. And this should include its distribution and also its general habitat and a host or substrates, for example. There is also need to look at some of the listed species among the macrofungi, and especially globally, regionally, and even at local scale. So that once you understand the description of the habitat for each of the species and its distribution and the habitat that it requires, then it is now possible to go to the next step. And the next step is what we are calling the species action plan. So species action plan should be for a selected threatened species, not for everyone. So a particular layer or very important or very endangered threatened species of macrofungi. And then under this, you also need to analyze the status of that particular macrofungi species so that you can be able now to lay down the species action plan for this particular species, which again should be monitored within what we call the species monitoring. So you monitor again at very specific scale and also at a broad scale. So you have both monitoring at the species or detailed species monitoring, and also broad scale species monitoring. We need also to understand the habitat approaches to conserving macrofungi species. So the first thing we need to do is to come up with macrofungi species that can be used as indicators of variable habitats. So we need indicators for woodland, for example, indicators of grassland habitats, indicators of macrofungi species in forested areas, indicators of macrofungi species, especially again in wetland and any other ecosystem, both terrestrial and aquatic ecosystem. Once we do this, then the next step is to gather knowledge of the extent and quality of now the available habitat types that are important for macrofungi. Again, like I've just said, all the way from terrestrial, the various terrestrial habitats, forest, woodland, savanna, grassland, for example. And then also understand the extent and quality of these habitats. Based on this information, it is then possible for us to go the next stage of identifying and conserving those important macrofungi areas. I'm calling them IFAs, the important fungi areas. We know that once you understand these important macrofungi areas, then you can develop management plans for them, for these sites, to ensure that these important macrofungi areas are developed. We can be able to borrow a leaf from other taxa like birds, which have already done the important birds areas. So this map shows the important fungi areas, obviously, and I'm only looking at some of the biodiversity hot spot areas in Kenya and in East Africa, and probably come up with an idea that we might again find the same areas as the important fungi areas. Again, there is need to do further research, and citizen scientists now have a lot of work to do and to help for us to see how such the important or hotspot areas can represent the important fungi areas, not only here in Kenya and East Africa, but both regionally and at a global scale. Now we are going to look at ex situ conservation of macrofungi. Ex situ conservation, especially of mushrooms or any species, refers to conservation outside the natural surrounding. So the species of fungi are collected and maintained, for example, in botanical gardens, in arboreta, or in spore banks. There are a number of steps that one must carry out in ex situ conservation. So one step that citizen scientists need to do is to establish establishment of a minimum target population to provide for the next 100 years. The other step is the distribution of founders through the various breeding programs. And lastly, implementing an overall plan that contributes to the objective of maintaining viable population across the globe. So this will start all the way from national, regional, and also at the global scale. There are a number of ex situ conservation systems that are available to our citizen scientists, or which our citizen scientists can apply. One of them is the spore bank. So you can be able to store spores in a spore bank, especially both for wild and cultivated gene pool. Another conservation or ex situ conservation system is the Botanical Garden, or sometimes people call them the mushroom garden. And this is very good, especially for taxonomy and research in Macrofungi. Pure culture collections, that is genetic resource collections, for example, and also the biological resource centers play a very key role in successful storage of fungal strains and should be emphasized in all countries, especially here in Africa. The objective of sustainable utilization of macrofungi requires that there is need for ensuring that utilization of ecosystem is sustainable, be it in grassland, woodland, or forest, or even farmland. There is need to encourage sustainable harvesting of macrofungi diversity, especially the edible mushrooms. There is also need to encourage good practice and sustainable collection of specimens, especially during scientific research. So when you are out there as a citizen scientist, you do not just collect anyhow. So we are encouraging you to follow good practice, that you do not just collect everything. You leave some of the mushrooms of the various macrofungi in the forest rather than collecting everything. There is a great gap in research on economics of macrofungi utilization. So we are encouraging local and international citizen scientists to carry out the research on economics of macrofungi utilization, because this is going to help quite a lot in conservation of macrofungi species. So on the issue of policy analysis and advocacy, especially with respect to macrofungi species, we are very aware that macrofungi species only rarely seem to be considered in national, regional, and even international conservation actions. And the policies at both the national, regional, and global level definitely lack issues to do with mushrooms, issues to do with macrofungi. And we need to review our policies so that they are also looking at the issues of macrofungi diversity, the issue of sustainable utilization of macrofungi species, the issue of management and conservation of macrofungi species. Therefore, that's why we are urging urgently that there is need to review, there is need to identify and validate and even adopt enabling policies, legal framework, and practices on macrofungi sustainability and conservation, all the way from national, all the way up to global scale or global level. Lastly, we need to understand that community-based conservation can make huge gains in macrofungi conservation. Therefore, community-conserved sites are critical complements to the formal protected area network. There is therefore need to build capacity for macrofungi conservation among local communities and also among the citizen scientists. We should also promote education and awareness about fungal diversity. And lastly, we need to establish all the competencies required in all stakeholders to carry out or facilitate macrofungi conservation. Thank you for listening and be blessed. Okay, thank you very much, everyone, for listening. Now what we are going to do, we have reached to the end of our presentation, but now we are going to question time. So I'm going to ask Dr. Mary Nyawira to take us through the various questions in the chat section. So in case you are there, you are the participant, we are requesting that you can write your question or your comment in the chat section, and then we are going to go through them and have answers. We have allowed 5 to 10 minutes, and then we call it a day. So let us have Dr. Mary taking us through the questions, and then the question will go to the various presenters. Thank you.
Okay, thank you very much. It's the time to go through the question. I can see we have several questions in our chat. We have one of our participants asking how they can join citizen science group because they are interested in joining this group so that they can be able to conserve the rock edible mushrooms. I think we have discussed 2 ways we can join this. Right now, we are trying to expand our monitoring program. So we'd like as many people as possible, wherever you are, you can contact us and we can form this monitoring program where this group of people can come together so that they can be collecting this data. And as we have been told, we have 2 methods. You can decide to bring your observation or your specimen to the National Museums of Kenya or any other research institution near you when you have collected them. You can also use the iNaturalist program and you upload your observation. You take a good quality photo and you upload it there, and we will be able to see that observation. We'll be able to identify it, and you'll be part of now the citizen scientist. So we have been told by Dr. Carlos how this program is expanding the science, or is making the science expand. So let us join the iNaturalist, and there we can upload our photos and we can be able to be of this big team. There is a second question where somebody is asking how they can know the edible mushroom. I think Susan can answer that one for us. Susan? As we wait for Susan, maybe Carlos, you can expand to these ones who are asking how to join the iNaturalist. How can they join as we wait for the next session?
Yes, actually, joining and using iNaturalist is no different from using any other social network. Remember, using iNaturalist is free. You don't have to be a professional photographer or have expensive equipment like cameras or expensive cell phones. Pretty much, if you have any kind of cell phone, you can use it to document any living organism, even dead organisms that you might find. Joining the iNaturalist community, you just have to go to, I'm gonna type this here, naturalist.org. You can just go to iNaturalist.org, and then you just can join in. You can use any of the social network that are already there to join in, or you can just use any regular email that you are using. And as beside of that, I mean, is joining the community is just having the intention and the will to do it. You know, one of the hardest part of being part of this community is to actually take photos and to document it, because believe it or not, sometimes user or people are like having like afraid or being ashamed, just stopping by any organism and actually taking a picture. That's one of the really difficult points of using iNaturalist, because we are not used sometimes to taking photographs of organisms. So we're going to see that more in detail in the next session. So I really hope to see you all there to learn a little bit more on how to use it. But if you haven't already been on iNaturalist, I really encourage all of you to go to iNaturalist.org and just create an account. You can do this also on your cell phone, and it's just go to the settings part of iNaturalist app and just join in and start a session. So it's not difficult. I really don't want to get too much into that detail, because we're gonna have a whole session for that in the next seminar. I hope this helps. So
thank you very much, Dr. Carlos, for that good answer. So those ones who are asking how to join the Citizen Science program, let's meet on the other week on Friday, that is 3rd September. We have a big session which will be discussing how to use the iNaturalist. Then now we are going to Susan to give us an answer on how we can identify edible mushroom. You can also include the effect of organic of pesticide and fertilizers on mushroom. Susan?
Hello, Mary. Can you hear me?
Yeah.
You can hear me?
Yes.
Yeah, so what I can say is that it's not very easy to identify which mushrooms are edible. And in most cases, we rely on people who have the experience on which mushrooms are edible. In Kenya, we have the termitomycetes, and these are mushrooms that grow in association with termites. And we find that some communities are conversant with these mushrooms. And so it is always important to rely on the knowledge of the people who know. And so in case the person who is asking, you are aware of people who actually, and people who are reliable to give information on which mushrooms are edible, then it is important to get that knowledge from the people who consume. Because in most cases, other than the scientific knowledge that we have, we rely on the knowledge that the local people have. And so the other thing is you can take a actually a photo of particular mushrooms, and you can send that photo to experts, and they can be able to tell you that, yes, this mushroom is edible, and that can be helpful. So if you have the contacts of the people that can help you, who have the expertise, and people who have the knowledge, it's good to post such photos. We also have like the mushrooms of Kenya Facebook page, whereby if you take a photo, you can post on that page, and people will tell you whether that mushroom is edible or it is not. Mary, is there any other question in that line?
There was a question on how the effect of fertilizer pesticide on mushroom community. Maybe you can expand that.
Okay. What happens is that when we use the chemicals and when you use the fertilizers in our land, we normally like, what happens is that mushrooms are organic decomposers, and they'll actually decompose any type of organic matter. And what happens is that when you use the fertilizers, or rather chemicals, they destroy the mycelium that is usually below the ground, because mushrooms are just not what we can be able to see, but we find that we have the extensive mycelium below the ground. And what happens is that these inorganic chemicals, they destroy that mycelium that is below the ground. And that is why when you use the organic matter, it is better because we have even different types of mushrooms colonizing the organic matter in form of manure. You'll find mushrooms that grow in manure. You'll find others that actually decompose very different components, which are of organic nature. I think that is all for now.
Thank you very much, Susan, for that detailed answer. We have, because of the time, we have another question which I'm going just to answer very briefly. There's somebody who is asking where they can get field guide for mushroom of Kenya. We are updating the current field guide we have, and we are hoping by next year this field guide will be out, and you can share your emails, your contacts, so that when we have this guide now, we can share with all of us. There is also someone asking how they can propagate mushroom spores so that they can get seeds for mushroom cultivation. In the next session, we'll be discussing a topic on how to cultivate mushroom from now the spores until we have the crop. So we are encouraging all of us to join that session so that we'll be together to discuss how we can now make these wild edible, these wild medicinal mushrooms, how we can bring them out so that they can be of a benefit to us, to our communities. So I think because of time, we are going to reach there. If you have any other question, you can send us through our email. We have shared the email in the chat. You can also contact us we are going to provide the telephone numbers. So feel free to ask any question, and we are there. All the presenters are there, and we are going to share everything that you desire to know about mushroom. Thank you.
Okay, so I think those were the questions. So in case you still have questions, you can always put them in the chat question. We'll be able to answer them even after this webinar. So for now, I think we are almost coming to the close of our today's webinar. But as we have said, there is the second series that will be mounted or it's scheduled for 3rd September. So we are requesting all of us, all our participants, please make sure that you are able to join by registering for the next session so that we can also continue. So I don't know, I want if there is any of our guests who would want to say something before now I make a quick thank you and remarks or thank you note. Carlos or Paul? Or maybe we have Telessa? Okay. Okay. Telessa, yes.
Hi, thank you, everyone. This was wonderful, and I just wanted to briefly say how happy National Geographic is to be a part of this fantastic project, and we really look forward to seeing how this develops and what's achieved as the Mushrooms of Kenya project continues to expand. So I'd like to just congratulate again all of the organizers and presenters for a fantastic job, and I look forward to the next session in a couple of weeks. Thank you.
Okay, thank you very much. Paul, is Paul there? Maybe something, and then a quick one. Just yes. Okay, Carlos.
I just want to really say thank you to all of you who were present today. And remember, this is a global community. Feel free to reach out to other members of this global community and really empower yourselves with this kind of knowledge. It's already there. It's really easy to use, and just again, an invitation. Join this community and start using it, because it's for all of us in every part of the world. Again, thank you so much for spending this time today here. See you soon.
Good. Thank you very much, Carlos. And now I want to take this opportunity, first and foremost, to thank our guest speakers, all of them. So on behalf of everyone here today, including on behalf of our participants and our presenters, I want to thank you very, very much for making time in your busy schedule to be able to speak to us today. Thank you very much. Also to our presenters, thank you for the enlightening and also very outstanding presentation that you have just presented to us. And I'm sure they have changed the way we know mushrooms, the way we know how to manage and even how to identify them and to deal with them. I want to take this opportunity also to thank our ICT personnel, and in particular, Mr. Nicholas Owino. Thank you very much, Nicholas, because you really helped us. Thank you for your tremendous support, and especially in organizing this. And now I also want to take this opportunity on behalf of the University of Nairobi, in the National Museums of Kenya, the National Geographic Society, and also the KENVO, that is a voluntary organization. I would like to pay my deepest and our deepest respect to all our participants, and to express our appreciation to you, our participants, for taking time out of your busy duties, because you have other duties, to attend this webinar. So I appreciate your keen interest in mushroom research and conservation, and I hope that what you have learned today and what you'll also continue learning next as webinar series will help you a lot as citizen scientists and also in your daily endeavors. So I hope that you will even acquire more skills and even have a better understanding of all this by joining, like we have just been requested, now our second webinar series, which is scheduled on 3rd, that is Friday in 2 weeks from now, 3rd September 2021, same time. And we are requesting you to join and register if you have not yet done so, our next series. So we will have the link in our chat. I hope Mary will put it. And it's already also in our flyer, but we're also going to send it to your emails, because we have the emails for you. But also we are asking if you have other friends to join, because we will continue now with the practicals, especially on iNaturalist tools in the cultivation of these mushrooms, for example, and also in looking at how you can contribute in the identification guide we are developing. So also note that we are going to present or to award certificate of participation for our participants who will be able to also join the second series. So please, those of us who are here, please make sure that you also do the same, so that you also receive this certificate of participation, which will be after the second webinar. Therefore, I would encourage you all to remain engaged as we have been requested. Let us continue submitting and also in the identification of our mushroom species, and especially through the iNaturalist. So please join us next webinar series, where we are going now to learn the tools and how to go about all this and how to use the iNaturalist tool, so that we can be able to continue contributing to our macrofungi research and conservation in our different ways. So I look forward, and we all look forward to welcome you once again on 3rd September 2021, that is 2 weeks from now for the final webinar series. So I'm sure in case you have any other queries, there is a project email. Mary, I'm sure you have put that in our chat section. That is marsproject2019@gmail.com. We also have our Facebook and our YouTube. The links are already in our chat section. I'm sure we are going to use that, and then we'll be able to continue. Otherwise, thank you all for your attention, and may God bless all of you. Otherwise, asanteni sana na Mungu awabariki. That means thank you very much and may God bless you. Thank you. Yes, I think we have come to our end of our webinar series today. So wishing you a very pleasant good evening. Thank you very much.